So this is absolutely fantastic. It's now the 29th of August 2013. It's about 1 o'clock in the morning and we're starting to get the egg sperm bundles released from this Acropora valida in captivity, southeast London at the Horniman Museum. So what Gary is doing is just pipetting egg sperm bundles that have been released. They've floated up to the surface, and we're going to take them down to the lab and show you how we're doing some of the microscope or some of the close-up photography of these egg sperm bundles. We're actually using red light to look at the coral here because you don't want to stress the coral out by using a regular white light torch. That could actually stop the coral spawning, and obviously that's the last thing we want to do. So we're using red light. It doesn't interfere with the spawning then, and it will allow the coral to carry on progressing, releasing the egg sperm bundles. So now we have the egg sperm bundles that we've just collected upstairs in the coral laboratory. And Gary's now pipetting those out of the beaker and putting them in these coin capsules, which we use then to take photographs of those egg sperm bundles. So we're using a Canon setup with an extreme macro lens attached to it, and what we'll do is we'll take some photos and just show you the egg sperm bundles when they're newly released, and then as they start breaking apart. It takes about half an hour for the enzymes to start breaking the eggs apart and then releasing the sperm package that's in the centre of those bundles. So when they're released, it depends on the species, but generally something like in the Acropora, something like 6 to 8 eggs are wrapped around a sperm package in the centre. And the reason why the eggs are around the outside is that eggs are full of lipids, fats. So as they're released out of the coral, they basically float up to the surface, and then there's that half an hour window where they're compact and stay together. They float up to the surface, and then as they break apart, that then releases the sperm in amongst the slick that's formed at the water surface, so the sperm can then cross-fertilise with eggs from other colonies. So it's an amazing evolutionary adaptation for fertilisation and increasing the gene pool of corals. So what I'll do now is I'll just quickly take a photograph. And we can see on the screen there the egg sperm bundles. There's a newly released one there, and then we'll cut in and show you one that's now releasing sperm. You can see the sperm right in the centre there is the white milky part of the image around those individual eggs, and then this image here, they're individual eggs that have broken apart from that egg sperm bundle.